I'm good.
You're good.
Do I have to start again?
You have
Happy
to start
New Year,
again.
everyone. Really excited to welcome our first speaker of the year in person. That's great. And before I introduce Ray, I just want to mention, I saw on your website that you consider yourself a physicist. Like to, paraphrasing, like to poke your nose into biology. And that's really what this ground is about. I'm myself a biologist who likes to maybe dip my feet in physics and computer science, so that's great. But it's great to have you. So Ray Mansbach is assistant professor in the Department of Physics at Concordia. And before you joined Concordia, you obtained your PhD in physics at the University of Illinois in Champaign-Urbana, and then did a postdoc at the Los Alamos National Laboratory in the theoretical division. And so your talk today is learning from life, no less than that. Lessons from machine learning and biophysical simulations. The floor is yours. Thank you.
Thank you for the lovely intro. I will say that I decided to go a little bit heavier on the machine learning today than on the biophysical simulations, but I will say a little bit about those as well. Before I really get started, I would like to begin by acknowledging that Concordia University, where most of this research was performed, is located on unceded indigenous lands. The Kanien'kehá:ka Nation is recognized as the custodians of the lands and waters on which the university stands, and Tiohtià:ke or Montreal is historically known as a gathering place for many First Nations. Today, it's home to a diverse population of indigenous and other peoples, and we respect the continued connections with the past, present, and future in our ongoing relationships with indigenous and other peoples within the Montreal community. And I just want to say that I do usually like to open with a territorial acknowledgement, but I also always want to remind people that they're a little bit controversial because sometimes they're used as kind of a box check. So I try to keep it in there to remind myself, you know, this is something I need to think about, and this is something that is not an endpoint in itself, but just a reminder of ways that we need to move forward. Hello, who am I? I'm Ray, or Dr. Mansbach. And one of the things I'm going to be saying a fair bit about today is that I always find it really important to remember that when we do science, we are scientists doing that science in context. So for that purpose, here are some of the things that I think about when I think about me, what's my context. So I'm coming from the perspective of being a scientist, but also being an author, an artist, a dancer, a friend, someone who's queer, and someone who probably has complex PTSD. So that's where I'm coming from. I bet other folks are coming from other places. I will come back to this and say a little bit more about it, assuming I have time at the end. So here's my lab. These I really want to highlight because these lovely folks are, of course, the people who did most of the research that I'm showcasing in this talk. They have been amazing. Just a really wonderful crowd of people. I'm really fortunate to have gotten to know them. So this is Reza, Tolawashe, Adam, Giler, Joe, Samit, Natalia, Zainab, and Brenda. And this is our lab poster, which again, this is a poster that's modified from a Creative Commons website with the purpose, again, of reminding, essentially, to summarize, reminding us that we are scientists who do science in context, and it's always important to me that everybody who wants to do science is able to do that science. So that's where I'm coming from, and what I'm now going to transition to talk a little bit about the machine learning side, primarily with a little smidgen of simulation. So I'm going to start us out with a joke from XKCD, which starting out my lectures with a joke is something I picked up from my dad, who is also a professor, although not in physics. But this joke is a joke, which is unfortunately a little bit true, which basically is talking about the fact that machine learning is the idea where you have an input to your system and an output to your system, and you let the machine figure out a solution that will get you from the input to the desired output. And if it doesn't look right, you just keep making it try until it comes up with a good solution. This can be a really good way of getting things done fast, but it's not so good if you want to understand what's going on, which is a problem that we're interested in my lab. Now, I would say sort of the biggest theme unifying the work we do in my lab, and I will give a very broad overview closer to the end, but the biggest theme that ties most of the work in my lab together is the development, design, and understanding of proteins and protein-based therapeutics. And we use two big areas of research. We use machine learning techniques and we use multi-scale modeling, primarily biophysical simulation, in the pursuit of, on the one hand, finding search spaces for these proteins and protein-based therapeutics and understanding and searching through these search spaces to find specific systems that are of interest for one reason or another. So today I'm going to be spending more time probably talking about the machine learning and the search spaces and searching through the spaces than I am on our more detailed investigations of the specific systems. So why would we want to do this? Well, machines are smart. We can use data-driven approaches, and probably we've all seen recently that these can be very powerful. They can be used in many different areas, from the creation of photorealistic images, like this generated image of a person, to designing artwork to specification, like this picture of an astronaut riding on a horse, to generating text, to designing drug molecules, to folding proteins, which is highlighted by the fact that AlphaFold 2 was recently honored with the Nobel Prize in Chemistry, to my personal favorite, which I'll say just a tiny bit more about in a bit, which is designing fluorescent peptides, which is a bit of research I was able to be involved in at the very end of my PhD. Now, on the other hand, machines are not always so smart. So, for example, you can have a machine that you've taught to be able to categorize different kinds of images, and you can show it this picture of a black and white bear-like thing, which you immediately recognize as a panda, and it says, yeah, I think I'm pretty sure that's a panda. But if you perturb this image with the right small noise, you can get the machine learning algorithm to be like, that is totally a given, right? And what this underlines is this thing is not thinking about these images, quote unquote, because these things don't think, but this is not processing these images the way that a person would process this image. And it can be very hard to figure out what it is doing, which is a problem, because when you don't understand what machine learning is doing, this can lead to performance degradation. So here are some examples of early attempts, not in my lab, but early attempts to generate drug molecules that fit into a particular receptor. And some of these algorithms are not very good at doing this off the cuff. They'll come up with things that are physically totally unrealistic and require post-processing. But without that post-processing, you're going to see the performance degradation. You can also see undetected failure. So if you don't know what's going on, maybe you don't know that it did something wrong. You can see misuse and out-of-scope application, like that time a couple years ago that some New York lawyers decided to use ChatGPT to write their legal briefs for them, which is a really bad idea. Because this is not, ChatGPT was not created with an understanding of legal briefs. It was created as a probability, basically a probability machine that can generate text that looks real. So this is all sort of an instantiation of a principle that's been around for much longer than we've been talking about artificial intelligence, at least, which is garbage in, garbage out. Garbage in, garbage out means if your data is biased, the data-driven model must necessarily also be biased. And this is a problem. It's a problem in terms of the kinds of things we're trying to do, because if you want to design a new protein or a new drug, your data is often biased by what people have looked at in the past. So it can be really, really hard to find something that's actually new, but still works for your use case. On the other hand, it can also have pretty serious ethical implications, because if you think, oh, well, a machine can't be biased, so I'm going to train that machine to do something like predict crime rates in specific neighborhoods, all your data is horrendously biased because in the past, people were biased, and therefore, all of the data on people who are getting arrested still has that bias in it. So it is always really important to remember this principle if you want to apply machine learning and to really understand your data. And I don't want to say that I've always done this perfectly. I would say that in some ways, this is definitely a process for my lab where we've been getting better and better at this. But it is something that I think is really important to mention and to keep in mind. So more specifically, the kinds of things that we're interested in my lab are using generative artificial intelligence to make and refine search spaces for drug design. So this is an area that I would say started in about 2018 with a paper from Alan Asparuh-Guzik's group at Toronto, where they used a particular deep learning algorithm called a variational autoencoder, which I'll explain to you a little bit more in a few slides, to basically create a map of small molecule drugs. And they showed that this map had some properties, which I'll talk about a little bit more in a minute, that allowed for them to optimize at least simple properties in that map. And therefore, the idea was, this was a very proof of concept, but the idea was you could use this map to optimize, to find new drugs, to generate new drugs for your particular use case. But then as I add here, you really want to understand the map, or your route planning could be kind of a disaster. This is very ongoing research. Now, I was fortunate enough to be involved in a project at the end of my PhD that built on this idea, but used it for a different use case. So in this case, what we were trying to do was design sequences of peptides or short proteins that would be able to aggregate into specific shapes. So basically, we wanted peptides that formed long and linear aggregates. So something like this one or this one that didn't not aggregate like this one and that didn't clump up too badly like this one. And what you can see here is the map that we were able to produce. Where the nice thing about this map is that we could really say, okay, if we don't want to be on this side of the map because we can tell that these are not the properties we want, it's much better for us to be on this side of the map, and we can generate new peptides to look at in greater detail, so that we can come up with the properties that we want. And that's unfortunately work that I'm no longer involved in because it's mostly my PhD supervisor's work, but it is ongoing. It's very cool. They're able to make these peptides that aggregate under changing pH to form basically little glowy nanowires, kind of. So they're good for sensors and stuff. Very cool. But what we're actually trying to apply it to now is the problem of antibiotic resistance. The bacteria really are bad, as clearly my computer agrees with me. We're seeing rising death tolls and dwindling pipelines of new drugs. So it's really important for us to be able to understand these things and to come up with search spaces so we can search more widely to prevent, here's my joke again, Albert from becoming a member of the antibiotic resistance. One possible solution to this is antimicrobial peptides, which are part of our innate immune system, but can potentially be designed to be even more to have even more efficacy than they do now. They're short proteins. Oh, let me get my laser pointer back. They're short proteins that have a higher affinity for bacterial cell membranes than for host cell membranes. And so they can often, many of them are capable of disrupting those host cell membranes, meaning that they can either be used to directly kill bacteria or to make it a lot easier for other drugs to kill those bacteria. And it's thought that the buildup of resistance to them may be slow because they attack the membrane in general. Having said that, resistance does still build up. So those are some things to keep in mind. But what we're trying to do in my lab is design antimicrobial peptides using this sort of big picture loop where we perform molecular dynamic simulations of particular peptides of interest, and then simultaneously actually, but use this information to help us build a deep learning associated search space that we can then use active learning techniques to search through as efficiently as possible, because it takes us a long time to perform these simulations or to ask our experimental collaborators to check how well a peptide works. So we really only want to be doing this if we know that something is likely to give us some useful information. Designing with deep learning like this is a subdomain of generative artificial intelligence, like I mentioned. Typically, the idea of what you're doing here is you are trying to train a model such that it can generate something that looks like your data, where your data is something that you have some idea of what it looks like to begin with. For some reason. For example, maybe you have a bunch of pictures of people and you want to train a deep learning model such that it can generate pictures that look like pictures of people. In other words, your training data, if you pick a random picture from your training data, you have some probability of getting some picture that looks like a person and a very low, basically no probability of getting something that looks like a horse if you don't have horses in your training data set. And you want to similarly have trained a model that generates things that could have probably come from the same distribution. But the question then becomes, how do you optimize this data for particular things, like in our case, antimicrobial resistance, and how do you actually understand the model? There isn't necessarily any kind of explicit mapping between where the model starts and what it gets out. So in our case, there are many solutions to this, but we wanted to use a model that has an explicit search space or an explicit map associated with it. So there is a machine learning algorithm called an autoencoder, which creates a map, but not a search space. So an autoencoder is an unsupervised approach to learn a compression, basically. The idea is if you start with, for example, an image that's described by 1,000 different values of a pixel, you then attempt to solve the problem of using a deep neural network to find a nonlinear function that maps from this 1,000-dimensional image down to a 2 to 5 dimensional image, or a vector of 2 to 5 numbers instead of 1,000 numbers of pixel values. The way that this works, the way that you actually do this, you find this compression, is by training a deep learning model to reconstruct the original data. So the idea is basically you start with your 1,000 pixel values of many different images, and you send it through one nonlinear function that takes it to 2 to 5 numbers, and then you send that 2 to 5 numbers through another nonlinear function that takes it back up to some 1,000 numbers of pixel values. The idea is, this forces the model to learn an informative mapping, because if it throws out all the information, it's never going to be able to reconstruct the original at all. For people who are comfortable with deep learning and similar, the loss function here is just the mean squared error between the input and the output. And what you end up with is you just end up with an embedding. So for any image, you can embedding or an encoding or a compression, you take an 8, it gets mapped to a point, just say a 2-dimensional point, a 9, it gets mapped to a 2-dimensional point, so does a 5. So these are all disjoint, disparate points. So that means that every input is mapped to exactly one output, and any space that doesn't have a point mapped to it doesn't have any information associated with it. It's basically just empty space, which is not what we want, because we want to be able to search through it. And you can't search through a bunch of disparate points very easily, because that would kind of be like trying to jump from one pillar to another pillar without any necessarily directional information. So we want to go from that to something that is more continuous, more smooth, and has something like a slope associated with it. Because when we have a slope, we have a good idea of how to find an optimum, because what we do is we just walk along the direction of the slope. In other words, you need something that's differentiable, and right now this isn't differentiable. So the solution to this problem is what are called variational autoencoders. Variational autoencoders, instead of taking every image and mapping it down to a single point, they kind of take it down and map it to a fuzzy region. So to a probability. In other words, your 8 stops going from a single point, and instead it goes down to a probability. And so if you are in your low dimensional space and you try to do a reconstruction from this point, you'll probably get something that looks like an 8, but you might not. As you get further away, you still might get something that looks kind of like an 8, but you might get something that starts to look like a 5. And what this essentially does is it overlaps the 5 and the 8 and forces this thing to come up with something that's continuous. It fills in those holes. Again, for folks who are maybe more comfortable with how deep learning works, the encoder is now trying to find a mapping. Sorry, the encoder is the first half, the decoder is the second half. The first half encodes, the second half decodes. The encoder is trying to find a probability distribution of the latent variable z given the input vector x, and the decoder is trying to find a probability distribution of the output, now the reconstructed x, given the input z. And those may not fully match anymore because there's this probability in here. And this is done by adding what's called a regularization term. If you see the word regularization, that just means we have an additional constraint beside the problem that we were originally trying to solve that we think is going to help us out. In this case, it's applying a prior to this distribution basically, again, it's just saying that you have to fill this thing out. But a lot of times it can also be used to, if you have more information, like more, you know, more about the physics or something, you can often bring this in as what's called a regularization term. So great. So we are trying to use variational autoencoders because they have this nice explicit map associated with them. So you're going to embed your data into this map. And now when you draw a point from the distribution of the map, you get a new data point that is probably related to the nearby data points. And what we were trying to do was construct search spaces for antimicrobial peptides. So we were trying to answer, or we're currently in the process of trying to answer the questions of what do these search spaces look like? How can we understand them so we understand what the model is doing to some extent? How do we use them to design antimicrobial peptides? And maybe more specifically, how do we search through them to find points that we're going to want to assess in greater detail? And so the first question, and to a slight extent the second, was a question that was addressed initially by my master's student, Sam Renaud, who graduated, oh gosh, a couple years ago now, I think, since we're in 2025. He was really interested in these variational autoencoders. And so what he did was he took a variational autoencoder, and now the inputs and outputs were actually sequences of amino acids, so the compositions of the peptides that it was trying to reconstruct. And what was added to this is kind of a standard procedure. This is almost like regularization in a sense again, but basically what you do is you've got your embedding, you've got your variational autoencoder, and you add to it, instead of just having a decoder as output from this latent space, you add a property predictor of some kind. So in other words, there's a neural network that takes your input to your map. There's a neural network that takes your map to your reconstructed input. If you take this point and if you take any point in this map and reconstruct it, you'll get some peptide. But you also add, as an additional problem for this thing to solve, can it predict whether one of these points is likely to be an antimicrobial peptide or not? By doing that, you're forcing it to consider the additional information, and this will change the map that you come up with because it'll actually pay attention to whether things are or not antimicrobial. So here's what he came up with. So these are maps for 5 different types of deep learning model architectures using different dimensionalities for each map, projected down into 2 dimensions. So, which I'll say a little bit more about in 2 slides. So each point is actually a sequence. The blue points are sequences of things that have not been verified experimentally to be antimicrobial, and the orange points are things that have been experimentally verified to be antimicrobial. And what you can see is that depending on the architecture and the dimensionality, these things may or may not be separating the antimicrobial from the non-antimicrobial portions. We can quantify that using something called the silhouette score. Basically, if the silhouette score is 1, that would mean orange group is separate from blue group. If it's 0, they're completely mixed up together. We don't actually want it to be 1 because then we've got disconnected things again. You can't search through them. But you can see that some of these things are doing better than 0, like the AAE, the transformer, and the WAE. And I'm not going to go into great details on what that means, but just suffice it to say those are different ways of coming up with these maps. We were also able to show, so these data points here are data points that we already had. The model had not seen them. They were not used for training, but they were existing sequences of real peptides that were or were not antimicrobial. Then we took the model and started generating sequences. Now, at this point, we were not able to do any experimental verification. We're sort of ongoing talking to our collaborators about this, but we did do a quick back of the envelope calculation by using someone else's deep learning model to predict whether the sequences that we generated were or were not antimicrobial. And the nice thing we saw was that as you went across basically here, for example, or here in just 1 dimension, the probability of the generated sequences being antimicrobial increased for many of these models. In other words, we are generating things in one location that have a higher probability than in the other location, and that probability tends to go up somewhat smoothly, which is what we would like. Now, having said that, I'm showing you all this in like 1 and 2D. I don't know if you can tell, but I have things like 32, 64, 128 up here. So the actual dimensions of these maps is still pretty high. These are projections of these maps done using principal components analysis, which is a linear mapping. And we did that because the nice thing about PCA is that you get a map down and you can then map right back up to the original space. And you can also know exactly how they are related because you get an explicit map between the original, wow, I'm saying map too much. You have an explicit function and an explicit inverse function, which is nice, but it's linear. So this can often distort the space that you're trying to project. And we said, okay, well, it'd be great if we could use this linear model to project these maps because we want to understand these maps. And if you can just come up with a linear projection into 2D, you can really trust what you're seeing in 2D if it's not distorted. So we actually quantified how distorted these projections were. Again, don't look at this in too much detail. 0 means distorted, 1 means it's great, nothing changed, for a few different ways of measuring distortion. But the thing I really want to point out is that, for example, if we compare the WAE to the transformer was doing a great job of separating these spaces. It was doing a great job of several other metrics that we use to measure its performance, but it's distorting the space more egregiously than some of the other models, particularly in this particular score, the cohesiveness score. So what this says to us is essentially for these architectures, the transformer is high performing, but it's not necessarily that interpretable because we have all these statements about this map that we can understand because we can look at it are less necessarily true. There's more distortion between this map and the higher space map. This is complex and we're still working on it, but it is a way that we were able to think about interpretability, in other words. I think we're running a little low on time, so I am going to just very briefly say we were also able to connect the latent spaces with basic peptide properties, including isoelectric point and charge and hydrophobicity, which is good because it tells us that the models there are some things about these models that we can understand because we also often measure the hydrophobicity of peptides and say this has something to do with whether it's antimicrobial or not. The model is also saying that. Now, one important question becomes how much data we actually need to be able to do this, to arrange the space, to get the blue and the orange separate, because we don't have a lot of data about many of these. So we have a lot of peptide sequences, but we don't necessarily know how many of them are actually truly antimicrobial. So those blue points, we just knew they hadn't been verified to be antimicrobial. We don't right now know they're definitely not antimicrobial. So it's a much smaller number that we have like explicit experimental data for. We would like to be able to use that explicit experimental data because it's much more, accurate. It gives us a much better idea of the map. The question becomes, do we need to have that data for every single point in the map to arrange the space? This is very preliminary work. The answer is no, fortunately for us. Using stuff that's easy to compute, my PhD student came up with, so here we again have these lower dimensional spaces that have been arranged by different variables. And then if we go down, so this is when we've labeled every single point. This is when we've only labeled 25% of the training data. We are still seeing that the space is doing a good job of arranging itself, even with only 25% of the data labeled. There's a lot of data, 25% is still actually a lot. So we're totting that down to see how low we can go before we lose that ability to arrange the space. But that's going well, which is exciting. We also want to answer the question of, okay, well, we've been thinking that it's important to arrange the space because we understand it better. We understand what we're searching through, but does it actually affect the search? How much does it affect the search? Because it matters if arranging the space in different ways changes how we move through the space to search it. And that is another very preliminary thing that we've been working on, that Giler's been working on here. And what you're seeing here is basically he came up with an objective function that he already knew the answer to, because right now we're just trying to solve this question for this problem. And he tried to optimize for this value by searching through the space in a specific manner using a specific algorithm. And what you're seeing here is how that objective function goes up as we search through the space for different arrangements of the space. And this is really, really, really preliminary. So the only thing I really want you to take away from this is that how you organize the space can change how quickly your value that you're trying to optimize for goes up. I don't want to claim that this is actually accurate yet, but that it is true that changing that arrangement can change how many times you have to evaluate the points that you're interested in. And this matters to us because remember, it takes us a long time to evaluate those points in great detail. So we've got our search spaces. We're working on searching through those search spaces. We're working on how to arrange those search spaces and how to understand those search spaces, but we would like to make those search spaces more accurate. So our next steps are to go, this is my horrible analogy. We currently have the world according to Hicatius in 500 BCE. There's some information there, but it's maybe not that accurate. And the reason that it's maybe not that accurate is we built this map using just the peptide sequences. There's no information on peptide structures. Peptides, firstly, can take on many different structures in solution. They don't just usually have one structure. They're so short that they take on an ensemble of different structures. And the structures that they have available will dictate their function. So this is important information. However, we absolutely cannot get information about, 100,000 peptide structural ensembles, but we showed before that maybe we don't need it. So what we're hoping to do is be able to bring in the information from running a bunch of short molecular dynamic simulations of different peptides, and by a bunch, I mean like maybe 1,000, we can probably get to reasonably. Bring that information in, pack it into our model as we did before using some of that other information. And then the hope is that we'll get to a model that's a map that's a little bit more reasonable, like this globe here that I don't remember when it's from, but probably not modern era cartography, but at least it's a globe, right? So that's the hope. That's where we're going. This is our big picture of this is one of our sort of driving questions. But I did also want to give you a flavor of kind of all the other things that are going on in my lab. So I'm just going to say a little bit about some of the specific systems that we're looking into. Not all of them are actually antimicrobial peptides, although we have been looking in great detail into one particular antimicrobial peptide that our experimental collaborators have been working with. It's called GL13K, and it's a fragment of a human salivary protein. And I think it's really cool because firstly, we know that it does puncture bacterial membranes. It does destabilize the membranes, but it also interacts with bacterial endotoxins. So there was a study that came out some years ago now that showed that if you inject mice with just the endotoxin, so not the actual living bacteria, you just inject them with the harmful toxin, well, their survival rate goes down. But if you also inject them with GL13K, their survival rate is much better. So not only is it doing something with the membrane, it's also doing something to interact specifically with the toxin. And this is something I think is really interesting because I think that in the future, it could have some implications on the resistance buildup problem, right? Because bacteria build up resistance because the chemicals kill them. If we can come up with something that instead of killing the bacteria just renders the toxins harmless, then maybe, that's another way to avoid resistance buildup. So this is very nascent something, but I did start thinking about it. We have been running molecular dynamic simulations to understand how GL13K aggregates under different conditions and how it interacts with different membranes to understand potentially how it protects against this toxin, that's the big picture question, and how it kills the bacteria. There's also work that I have ongoing that's a little further afield, but still proteins, where I have been working on photosynthetic proteins with a collaborator in the physics department at Concordia. This is work that was done by our co-supervised master's student, where she ran molecular dynamic simulations and was able to find the tiny molecular level origins of some experimental signatures, which really were able to validate a particular theory in that field, which was pretty cool. Another thing that we think is really interesting, so here we are back in proteins again, is so turns out toxin peptides are really cool on their own. They can be used for therapeutic applications. There is, in fact, an analgesic on the market that's derived from the venom of an aquatic cone snail. So they're really interesting for therapeutic reasons because they interact with neuromuscular receptors, basically. But toxin peptides tend to have a lot of cysteines, and they tend to have covalent disulfide bonds that restrict the shapes they can take on, but are also hard to simulate. So we have one student is looking in detail into the different ways that these things fold. And this is not something that I ever thought I would get to say before, like last year, but did you know that mucus is really cool? Did you know that mucus is also primarily composed of disulfide rich proteins? So this is a bigger scale thing, but it's similar to these toxins because they both use disulfide bonds to hook things together, basically. It's really interesting because, well, for a number of reasons, but one of the reasons it's interesting is because it has a lot of interesting viscoelastic properties and can be used as a base to kind of come up with new biomimetic materials, which I think is really neat. So we're working on modeling mucus, basically, which is cool. And we also have a couple of other pieces of work that are a little bit further afield due to the interests of my collaborators or my students. I have one student who's working on designing small molecule antibiotics that interact with RNA. And I have another student who is working on using network theory and deep learning to try to answer questions about MRI images in the context of how long COVID affects the brain. So this is all really cool stuff. And I just really wanted to give you a little flavor for it. And I think, yeah, we have a little bit more time because I think I zoomed through the middle there, fortunately or unfortunately. So I'm just going to sort of close with a few more sort of general considerations for grad students and for other people who are getting into science, because it's been important for me. I start here by asking the question of how the heck did I get here? Because I was born in the middle of the US in a tiny university town in the middle of farmland, and I've ended up in Montreal somehow. And the answer is not in a straightforward way. I went and did my undergraduate in physics at a college in Pennsylvania near the coast. And then I was like, I have no idea what I actually want to do with my life. So I'm going to apply to a bunch of PhD programs. And I got into a PhD program in robotics. So I went to Pittsburgh. I was like, this will be great. And I lasted for like 2 months before being like, this is not great. I hate this. Get me out of here. It was not a good match for me. And it was not a program that had a lot, I would say, of infrastructural support for minorities. They were very proud of the fact that the first ever PhD in robotics was earned by a woman, but there were, in my class of 15, 20 people, there were 2 women and me who's gender fluid. So something still kind of going on there that was screwy. I hated it. I was miserable. I didn't know what to do. Now, this is ridiculous, but I had also gotten in, when I applied to these programs, to the PhD program in physics at the University of Illinois at Urbana-Champaign. Now, I turned them down, and I was like, but they seemed really nice, basically. So I called their graduate department director, sobbing from a parking garage in Pittsburgh, like, help, I don't know what to do. I turned you guys down, and I'm just, I am totally lost. And he was like, it's fine. Reapply, you will get in again. It will be completely okay. You can come here. Like this man is so busy. As a prof now, I'm just like, wow, that is ridiculous that he took 10, 15 minutes to calm down a crying prospective student in a parking garage. But I did. I went there and it was a lot better. UIUC, for a number of reasons, just turned out much better. I rattled around a little while as I still tried to figure out what kind of research I wanted to do. But I finally ended up in biophysics, which was very, very fun, computational biophysics specifically. And then I went to do a postdoc at Los Alamos National Lab, which is where I got interested in conotoxins and was doing a lot of work on conotoxins when the pandemic hit. The pandemic hit like right after I signed my offer letter to Concordia, like a month after, which was super, super exciting. And I don't recommend ever starting a faculty position in the middle of a pandemic. Still has repercussions this far on. But fortunately, so I had to do emergency research, right? I was doing COVID research all of a sudden because biophysics, the theoretical biology department. And fortunately, my spouse, who is amazing, did like all the logistics to get us here. We drove from New Mexico to Montreal in 3 days, and we lost his ADHD meds in Texas, which I also don't recommend. The point that I'm trying to make here is that it is not as straightforward as it looks, no matter what. No matter how straightforward you think it should be, it's not going to be that straightforward. You're going to make a lot of screw ups. You're going to make a lot of mistakes. This is a very incomplete list of mistakes that I came up with that fit on the page from the last decade, including things like last term, I spelled my department chair's name wrong in my tenure package application, which is really embarrassing. I have also failed a lot. You will also probably fail a lot. This goes back even further than a decade. And again, there's more that doesn't make it in there. And I just feel like we don't talk a lot about the mistakes we've made. We don't talk as much about the things that we haven't succeeded at. So I like to highlight that a little bit and just say, if you're like me and you struggle with the idea that you are a person and you have value outside of science, and just because you're you are. Here's my favorite quote. Well, one of my favorite quotes that my spouse constantly says in my ear: "It is possible to commit no mistakes and still lose. That is not a weakness. That is life." I'm a huge Trekkie, although I will actually admit to not having seen that much of The Next Generation. But I really like this quote that Picard says to his trying to become human robot Data, who is, yes, named Data because, I don't know, we were very creative at that point. But anyways, I think that's really important. I'm just going to close with an acknowledgement slide, which is maybe a little bit less the normal kind of acknowledgement slide. Of course, I've already showed you my students who, once again, they did all the research, right? So big thank you to them. And of course, thank you to my funding and my computational resources. But I just also have a scattering on here. This is by no means complete, of various people who helped me. Some of these are my collaborators. Many of them started out as this is Sri. She, we are collaborating now, but she was my co-postdoc at Los Alamos. So we went through a lot together, especially during the pandemic when I was closeted in my bedroom and she'd already moved to the States while still trying to do her postdoc. It was crazy. This is my PhD advisor, Andy. This is my postdoc supervisor. But then we also have people like, oh, this is Lance. This is the guy who comforted me for 10 minutes and when I was sobbing in the parking garage. He's amazing. These are a couple of my undergrad professors. This is a prof at UIUC who, when I said I actually don't like working in your lab, I'm so sorry, she was like, that's cool. Did you want me to be on your committee anyways, even though I'm in condensed matter and you're in biophysics? And I was like, yeah, that's very sweet of you. Thank you. And let's see. This is a friend who's super cool, who works at Rochester and with whom I squeal about a lot of nerdy things, occasionally, like Ace Attorney and similar. And this is my friend Elizabeth, who got her PhD 2 years before I did, and one of the only reasons I was like, well, somebody managed it, so apparently it's possible. My point here is that this is not a one-person endeavor. This has been a very big network of support endeavor, and I just want to thank everybody, and I'm running low on time, so I want to thank all of you for listening, and I'm happy to take questions.
Questions?
No.
I'll start with one. When you were describing like the blue dots and the orange dots, and you said that the blue dots are not all confirmed that they aren't antimicrobial, you just don't know. Why don't you have 3 categories of confirmed no's and unknown?
Yeah, that's a great question. And the answer is this is one of the things that we really should have been paying more attention to our data when we did it the first time. Because the first time we did it, we were thinking about antimicrobial, non-antimicrobial. And we had a really small pool, and the reviewers were like, what about your negatives? Are they actually confirmed? And we were like, no. So yeah, actually, that's something we're considering doing in the future,
Okay.
but it literally hadn't occurred to us. This does work, but it would work a lot better if we had been more careful with our data.
Two things, one, a comment and two, a question. One, a comment, I strongly recommend Next Generation, it's really good. And in terms of a question, can you talk a bit more about the distortions caused by PCA?
Yeah, sure. Let's see. So basically, we measured these distortions using these metrics from this 2021 paper. Trustworthiness, continuity, steadiness, and cohesiveness, which essentially measure how much you, let's see. So trustworthiness basically says we split up two neighbors. Continuity says we made things neighbors that weren't neighbors before. Steadiness and cohesiveness are kind of similar, but for larger, like groups. So essentially, it's a measure of how much you're mixing up things that somehow weren't close, or you're spreading things apart that weren't too close in the original sets. And all of these run from 0 to 1. I haven't, to be honest, I haven't looked at the exact details in a while, which is why part of the reason, also we're running a long time, that I pulled out the sort of big picture here. But you can go through and actually think about what does this mean for your space. But the one that we really saw tanking out was the cohesiveness in the transformer, which says it's squishing things too hard. In general, PCA is known, now, again, these are one set of ways that you can measure this, but there are many different things that might also go wrong, and I don't have the slide on me, unfortunately. But basically, if you have something that's nonlinear, PCA is going to draw a line through it, and it's just going to squish things on top of those things, and that can be more or less of a problem. I still really like PCA because of the interpretability angle, but I do also like thinking of ways to quantify how much it's distorting the space. Having said that, we're probably, this may not be every way it could distort the space.
Thank you. I use PCA a bit in my work, and I'm about to use it a bit more. And I had never heard about these distortions, so it's really interesting to hear about them.
You should be able to pull up that paper if you just Google Geom 2021 and those metrics. But if you can't, you can get hold of me and I'll find it for you.
Perfect. Thank you very much.
I wondered why, I know you said like using linear, you can you have like your function and your inverse function. But is there any benefit to using something nonlinear?
Yeah, it's not linear.
Yeah,
It's
yeah.
more, I mean, the thing is really, so what we're already doing is nonlinear mapping,
Yeah,
right? If we add another nonlinear mapping in there. Okay, so in the field, I feel like it's very common. I'm actually going to pull back slightly, sorry. In the field of variational autoencoders, deep learning, and this kind of stuff, I feel like it's very common to say, here is our projection of our space, and then draw conclusions based on that projection of the space, which is something that I am sometimes a little uncomfortable about. I like PCA because I know exactly what it's doing to the space. Most other actually projection algorithms, you don't. You could train yet another neural network or like another variational autoencoder to maybe do it again. I don't know if you would do any better, and you would just run into sort of the same recurring problem. So I feel like it's an issue of like, we really want to be lower dimensional, but how low dimensional can you get? At what point does it help to just cut the corners with PCA?
Question about the models that you're using in simulation. So is there a whole sort of domain of understanding or interpretation that comes from playing with the model itself, assumption controlling for some of the mechanisms you put?
Not yet, but some of the
Yeah.
things we have, like different types of peptides, for example, we haven't really looked into much,
Mhm.
but it's something we're considering putting in. So similar to how.
Yeah, then I guess using those models as experiments, right? Because you can turn off certain mechanisms, you can
Oh, you mean like when we're actually doing the simulation?
Exactly.
Oh, yeah.
So then you
Okay.
can generate whole sets of data
Yeah.
that include some mechanisms or.
The hard thing is that the mechanisms tend to be. It's really hard to actually simulate the destruction of a membrane. It takes forever. So we keep having to run, even with doing stuff like this, we keep having to run into proxies on proxies on proxies. So that is something that we're looking into. So right now, Giler is looking into whether he can proxy membrane destruction with interaction with specific lipids, for example.
And I would say if there are people online who have questions, you can raise your hand or put that in the chat.
I have one more question. When you were talking about the next steps of not just using primary sequence, but also doing the MD simulations, what would your input data look like? Because you're going from something that's sort of one-dimensional to like, is it all of the distances between all of the amino acids? And also if there's multiple conformations, then is it like three-dimensional?
There's more than one way you could do this. So the sort of the quick and dirty thing I was thinking about doing just to try proof of concept was literally just take percentage of time in particular secondary structure for amino acids. But there's a whole lot of literature out there on deep learning models for peptide configurations. And they do sometimes look at things like you were saying, like distance between different amino acids at different times, or maybe average distance. I wanted to do something simple. Basically, I'm like, I'm gonna do something simple. Hopefully it won't be simple, but we hope, and then try to go to something more complicated like that. Yeah.
Yeah, I was just thinking the size of the input.
Yeah,
is gonna grow.
it's true.
Yeah.
It's a problem. And that's, yeah.
You had a question?
I was just wondering if you could briefly go over again the bit that you were talking about, like the representing the polypeptides as their primary sequence versus their structure. I don't think I followed that.
Well, I also didn't say very much about it.
Yeah, I feel like based on like, my like bio 200 knowledge, like
Yeah.
sequence should dictate the structure.
Yes,
Pretty
in
strongly.
principle, yeah. So in principle, sequence should contain all the information that you should need in a perfect world. I agree. The thing is that with the machine learning algorithm, it actually, the more information you can give it that's specific to your problem, the easier it is for it to find a solution. So in other words, if you give it the sequences and you have infinite time, you probably don't need the structures. But we don't have infinite time, so to find a better solution, the more specific information, without having to get it the map, right? It would have to figure out, okay, well, how does this do this? How does this affect this? So then the idea is basically, okay, we're going to cut out the middleman a little bit and give it some more information to push it towards the solution. Question of time.
A follow-up question.
Yeah.
Briefly, sorry, don't apologize, but I guess my next question was going to be like, given that information is like, if you have like multiple structures that are like very similar but have very small differences, say like the side chains that just like a little bit of a different angle, but it does functionally the same thing. Like having all of those structures seems like you might be like having a lot of very redundant information and it may not be efficient. And the model could kind of shortcut that by just recognizing that these sequences form this classing structure and will interact with the membrane in this way. But then you have to balance that with giving it the actual information. I was just wondering what, if you looked at the different ways that you could embed your data and actually give it to the model, and if you found that that has like a really significant way, like impact on the performance, and which ways of embedding your data.
Well, I don't know yet. That's very much I would say, an active area of research. But yeah, I think one of the problems is actually what you pointed out is that certain embeddings are more informative, but they're bigger. And if you get too big, you run into computation time issues again. So there really is that balance. I agree.
So just following up on Sean's question, is it, so from what I understand, your peptides are small enough that we should have, assuming obviously pH and all of those are constant, like the structures for them already. Is it not possible for us to just throw in those, or, fairly strongly predicted structures for a lot of these peptides? Is it not possible just to throw these in entirely and skip, like, because from what I understand, you're trying to like, reach a middle ground, right? Between the sequence and the structure by using, hydrophobicity, polarity, et cetera. But
So yes and no, I think is
Okay.
the answer to your question.
Okay.
First of all, we are actually currently looking at how well things like AlphaFold predict peptide structures.
Mm.
The answer is probably fairly well, if you make the assumption that there is a single native structure. And that's actually, so unfortunately for antimicrobial peptides specifically, this is that you can't just be like, well, given that the pH, given that the this is all the same, because what often happens is that they'll get structured at the membrane. Like there will be that change in structure, and oftentimes we don't think about that. We don't necessarily even have the structure at the membrane, we just have the structure in a different environment. So this is something we're really thinking about very hard. Yes, it is potentially possible. We certainly could give it some structure, right? For certain things, we could predict a bunch of things with AlphaFold, we could get it to learn that. And that is a line that we are investigating, but we also know that the environment does matter, that AlphaFold doesn't know things about the environment, and that structural ensembles may actually be very relevant, and peptides aren't proteins, so whether you actually want to think about them as having a single native structure at all is kind of a question mark.
Sorry, and follow up to that.
Yeah.
This might be a more naive question, but do antimicrobial peptides, like obviously if we were to inject them, for example, into a patient or give them orally, like they would be structured. So do they denature and then like reassemble in the membrane?
So a lot of antimicrobial peptides, it's actually kind of the other way around. In solution, they're just unstructured. It's not that like they don't really nature and denature the way big proteins do.
Right.
They're often kind of random coil, which is one of the problems. So they're maybe a little bit closer to intrinsically disordered proteins.
Oh, okay.
So if you like inject them and they get close to the bacterial membrane, oftentimes, so for example, GL13K, that we've been talking about, yeah. So it's forming okay, so here, for example, you can see it forms beta sheets when it's interacting with itself under specific pH conditions. Here it's forming more alpha helices near the membrane, but absent the membrane, it's probably running around as a random coil.
Oh, okay.
Yeah.
Like.
I don't see any comment in the chat, so thank you again for the great talk, thanks.